All right. I hope you all enjoyed lunch.
Thank you.
We're gonna have a session for another 3 hours and then we're gonna walk over the herbarium, but let me first introduce DNR's Chief of Staff, Shannon. Who's gonna say a few words.
Hi, thank you for having me. Director Boyle's had a death in the family, so he's in California, but otherwise he would be here to welcome y'all today. We are thrilled to have y'all. I wore my botany dress today, because I'm here speaking to you guys with all my flowers on it. Herrick Brown and I have known each other for many, many years. Known him since I was about 5 years old. I told someone today that I was surprised Herrick became a botanist because I would have thought he'd be a snake and reptile from all I knew about him growing up, but this is a close second. So we're thrilled y'all are here. Our botany team under Keith Bradley is the strongest it's ever been. We are just thrilled where it's going and all the things that he's doing. I saw Czilla and Hannah. Where is Hannah? Yes, out. I was on a Saturday at Table Rock about to hike up with my family, and I see them there taking photos of lichen on the side of the river. And I said, Czilla, are you doing that for DNR on a Saturday? She said, yeah, I've just found this wonderful thing. So it's just fun to watch y'all work and fun, fun to see where this group goes. But we can't do what we do without y'all's help. You are the reason we can store things and have history and, find out where things are and should be and give us our history and as well as keeping our pieces for the future. So we're glad you're here. I hope this collaboration and networking continues. I know this is the first time y'all been in one room together, probably ever, or, in this group. So we're thrilled y'all are here. We hope this only leads to more and more collaboration and more communication and giving y'all the support you need to help us do the work that we do. So we appreciate y'all being here. I'm going to stay for a little bit. I've got a meeting at 2, but I do want to see what Keith and Czilla have to say. So thanks again.
All right, for this talk, we're gonna be tag teaming. It's gonna be me and Czilla, Herrick and Czilla and me. So we're gonna be going back and forth, so you'll be ready. Hopefully we won't go over either. Czilla can't yell at herself to go. So I want to talk about, this is a big overview about the collaboration that has been going on since the 1970s between DNR and the work with Herbaria and the data we process and the data we use to drive conservation in South Carolina. So the introduction to the Heritage Trust Program, we are fortunate to be, we have a long history with conservation in South Carolina through the Heritage Trust Program. The Botany and Plant Conservation Program has existed since the 1970s. It is under the Natural Heritage Trust Program, which is a bigger umbrella that includes other divisions of South Carolina DNR, including Marine Resources Division, the Wildlife and Freshwater Fisheries Division, and a different part of the Land Water Conservation Division, which work here, including the Archaeology Program. The history goes back to 1974 when the program was created by the Nature Conservancy. You can see all of the people that have been influenced by botanists like Wade Batson and the University of South Carolina, like Doug Raynard, who was our first botanist. Chick Gaddy was actually the first contract botanist for the program. John Nelson, who's still in the room, foundational ecologist with DNR, hired Kathy Boyle after Doug Raynard left. Then we lost Dr. John, but he went on to great things as well. Bert Pittman started in 1990. He retired at the same time Joe Lamaris, who's our data manager in a different division. We were probably interrupting his lunch downstairs with all of our noise. He introduced us to the era of Esri, changing the way our data systems work. Kathy Boyle was our ecologist for a long time. She retired. Then I started in February 2020, which was days before COVID. Before people knew I existed, and so I didn't have email for a full year. And then there was a chain of hiring great people, hiring Samantha Tessel as our upstate botanist, hiring Czilla, who eventually became our full-time botany data manager. Eric Ungberg is our low country botanist, and of course, other technicians that we've got online, like Hannah and Michaela, Martin, fabulous people. So we're getting a lot done with this great talent. And it would be great talent if this thing would work. Oh, there it is. So like I said, this is our 50th anniversary, so we're very proud of the program. It was started by the Nature Conservancy, and when it was created, it was spun off to other states around the country. So we were the first in a line of driving statewide conservation actions around the state to buy lands for conservation. That's the big goal of the Heritage Trust Program, to buy properties that are valued for their natural resources, but we're a little bit unique in the state heritage program network, and then we also buy properties that are important for cultural resource preservation. So things, places like Poinsett Bridge, we've got properties across the state from the coast to the mountains. So our goal of the plant conservation program is to manage data to drive conservation in these land acquisitions. We track plant species, S1, S2, and S3 species. So I'll talk about that in a moment. It's too fast for me.
I like it.
It's doing it on
Auto?
We collect data from herbarium specimens, like naturalists, published materials, word of mouth. And we have an existing database. It's now 50 years old of legacy data that we're also improving with use of herbarium specimens. We create these classifications with nature cert criteria, S1, S2, S3, SX, and SH. And we
I like teeth.
It's trying to tell you, you only have so much time.
I'm being pushed here. And a lot of these plant species are representatives of imperiled ecosystems as well. We track certain things that are important to wildlife species, like milkweed species like the Asclepias that are important for monarch butterflies, which are under Endangered Species Act listing consideration. Okay, move on, I guess.
Now I want to.
Is there any way to like see my titles at all?
Yeah, that's sorry.
Never do the wrong thing.
I once gave a talk where none of my images came up and the whole talk was just images.
Oh, yeah, but disappears. I apologize.
So this is a big partnership that's been going on for the history of DNR, of working with the University of South Carolina's Herbaria. We've been collaborating for decades. It's been integral to everything we've done, and it's a lovely path through the forest. 2024.
Should be better now.
Thanks. Okay. And so that's just an introduction. I will be back to the kinds of work we do, but now I'm going to hand it over to Herrick. So that's the introduction to the collaboration. Yeah, it'll hopefully not do it automatically. Exactly. There is a laser.
Yeah, okay, so a very brief history at warp speed. South Carolina has been kind of on the botanical radar for hundreds of years. There are vouchers in European herbaria that predate Linnaeus. Think of Catesby and Lawson in the Carolinas. So their collections are available in some of those institutions. And so our legacy of botanical investigation dates back at least 300 years. And some of the earliest herbaria that are still extant in the state are those associated with the late 18th and early 19th century. So think of like the Elliott Herbarium at Charleston, or the bulk of the Ravenel material is now curated here at USCH, still outright belongs to Commerce College, but we take care of it here. We saw the map earlier with the barbecue regions and the 16 herbaria that we are aware of in the state. And so we're still trying to kind of figure out where all those are. And I think we have at least 10 or 11 folks representing some of those institutions today. And then the history of Herbaria and SERNEC, and this is kind of starting to speak towards our efforts towards digitizing the information that the collections hold. SERNEC was originally sort of a loose-knit group of curators that would meet annually at the Association of Southeastern Biologists, which is a regional biological society. SERNEC's, the original concept was not limited to herbaria. So that's the acronym there is the Southeast Regional Network of Expertise expertise in collections. And at that point in time, we were thinking like all taxa. After a while, at these meetings at ASB, eventually we formed something called the Society for Herbarium Curators, sort of more formalized this regional consortium of Herbaria. And then Zach Merle at Appalachian State University put up a proposal to NSF to fund a research coordination network that really kind of formalized the efforts for the Southeastern Herbarium curators to meet and further develop SERNEC and come up with a plan. About that same time, this would be the early 2000s, Specify was this sort of database that was on the horizon. Most of us, if we had the equipment and software to start cataloging collections, we would use something called Index Kentuckiensis, which was really just a Microsoft Access driven database. And when I moved back here in 2007, Index Kentuckiensis was great. It was doing good things. But it was developed in-house by this retired fellow who was, I think it was like a mechanical engineer or something that knew Ron Jones at Eastern Kentucky University. And so he was retired. There wasn't much support for Index Kentuckiensis, and the Specify system was kind of on the scene. And my understanding is that Carol Ann might be able to speak to this, that NCU was one of the first herbarium collections to use Specify, which was originally built for fish collections at Kansas University. Anyway, so they served as a little bit of a guinea pig for Specify. And that carried a lot of the SERNEC consortium forward for a few years. And eventually we learned about this other system called Symbiota, which is really what drives the SERNEC portal now. And so at any rate, I'll back up. The Research Coordination Network eventually pushed forth with a proposal to NSF to fund a thematic collections network. These were projects that were funded through NSF's Advancing the Digitization of Biodiversity Collections program, which is now sunsetted. It sort of transmorphed into something else. But we had at least 3 attempts at putting in a proposal before it was successfully funded in 2014, and it was a very large project. It involved upwards of 90 herbaria across 14 states in the Southeast. It was a very coordinated effort in terms of we had state lead principal investigators that would submit proposals, and then we'd have subawards to other institutions within each of those states. And so each state moving forward really had kind of a different game plan. As far as how this translates into the Society for Herbarium Curators, basically that group was meeting at Botany meetings, which is a more national sort of forum, and since SHC at the time was just kind of Southeastern curators, they said, well, here, this was Dr. Herr's kind of mechanizations or whatever, thinking outside the box. Well, he said SHC should be a global organization, and SERNEC essentially becomes the first regional chapter. So that's kind of the nature of that beast there. As I said, the project was funded. There's the award number, and the slides are live. You can read straight through if you want to read more of the history. Originally 90 herbaria included. I think our goal was to digitize 2-point-something million specimens. But once we started moving forward, this thing became somewhat of a juggernaut, and it just acted as a catalyst to like bring in these other little collections that had not the means to digitize stuff. They didn't have a computer, they didn't have software, whatever. And the Symbiota environment that runs the SERNEC portal today is more of a distributed sort of system where you don't have to have that upfront costs and investment in order to start entering data. You can just log into the website and start cataloging your collection. The original funding was for 4 years. It got extended at 1 additional year, and then through the addition of 3 partner and existing network grants, the funding actually ran through 2021. Okay, so in a parallel universe, as herbaria are going forward and trying to digitize these collections, and again, the goal of that SERNEC project was to daylight these dark data. If you think about the physical collections, they're typically filed away in these cabinets. We'll see them in the tour later. Often they have doors, sometimes they're, open air. But it's trying to find all that valuable information that's in the collection. It involves a lot of time and painstaking effort, just kind of what we call doing a herbarium crawl, pulling sheets, looking at them, seeing if it's something that fits your project or your research needs. And so in a parallel universe, 1974, at the SC legislature, we've heard all this a couple of times already, that essentially formed the Heritage Trust program through the Heritage Trust Act. And that essentially charged the personnel involved in that program with identifying natural or cultural features that were present in the state and then working to preserve them. So part of that is like recognizing, well, what do we already know? And the natural history folks that were involved in that program at the time were like, well, we've got all these wonderful natural history collections and they go back 300 years. And so folks like Doug Rayner and maybe Dr. Nelson over there visited places like New York Botanical Garden and other places to just kind of do these herbarium crawls and run through the specimen sheets and say, oh, this is a new one for the state. We need, there's only one record of it. So they prioritized those species based on how many vouchers they were able to find. So now what's happened is that these universes have collided. SERNEC has grown. As I said earlier, we added, I think we're up to about 130 collections in the portal now with roughly about 5 million specimen records. And regionally, we estimate there's like 233 collections. So there's probably a lot more out there across the Southeast. And so data access is no longer limited to these physical specimen crawls. It moves at a much faster pace. The data are parsed in a standardized manner, making them much more easily discoverable. And what I mean by that is that the county goes into a county field. And so you can sort all your records based on that one little scrap of information. And if you think about how physical collections are organized, they're usually taxonomically arranged with maybe some general geographic way of coding the way things are filed away. But in the digital environment, you can sort things in a much more broad variety of ways. And so this has kind of led us to discovering much more rapidly the species that are priority, previously undocumented records that are things that DNR are tracking, new introductions to the states or the Southeast region turn up more quickly now. And what we found is that through this partnership with Department of Natural Resources is that the way they're using these data is actually improving the quality of data that we conserve. We get feedback from it. We get annotations all the time. And sometimes it's like a day or a week of work for somebody like Avery that's going through and trying to deal with all of Eric's annotations that he's applied digitally. But that updates our taxonomy. It changes the priorities in terms of like our understanding of how many species are present in the state or if they're not. And I'll talk more about that in a few more slides. Csilla.
So talking more about universal slide, I was just going to talk to you guys a little bit. How are we processing our data? So you guys are working really hard. You're digitizing, you're putting this data up on SERNEC. What do we do with it? So as Keith talked about earlier, our primary focus are the tracked species. So those are the S1, S2, S3, SX, or SH species. SX is extirpated, SH is historical. Those are always fun ones because they usually have handwritten labels and they're really hard to read. But what we're looking for is an occurrence. So it's an observation of the population at a unique date and location. And a record is a database, whether physical or digital representation of all or part of the occurrence. So there are different types of records that we deal with when we process data here. There are observations. So that's the most common. That's when Eric or Sam or Keith or somebody goes out in the field and says, okay, I saw it, and they take notes on it. There could be surveys, we get phone calls, we get text messages, emails reporting these plants. There are specimens, which are the herbarium specimens. And then there are just images. And then there are a couple of combinations of all three of these. So iNaturalist will have a photo, but it will also have commentary on the actual population. And some of the specimens have either image specimen, like image of the specimen, or they will have in situ attached. That is becoming more common. A lot of curators are trying to encourage people, collectors, if they have in situ or habitat photos, to attach those to SERNEC records. We do have that ability to upload that onto SERNEC and look at it. So it comes, so the ideal record is where you have occurrence information, specimen information, the image and the photographer information, and location information. So there's a problem here, which is that database portals are really bad at mapping, and mapping portals are really bad at data. And then in the future, we're gonna add more stuff like micrographs, stack focus, as Mihai talked about, DNA, which is another separate portal. And UV imagery is just an example, but there are all sorts of things. So we are an aggregator that are dealing with a kind of mishmash. We look at a bunch of different portals. So here's an example. We can have a Clemson record that has an annotation, but the USCH record doesn't have that annotation. There could be cited specimen information on one particular specimen, but not on the duplicate. So that's information if the specimen was used in a paper. And why do we care about that? Because sometimes the scientific papers have a much more detailed list of the associated taxa. They sometimes have a map or they have other information. The iNaturalist record, which is very common, that's now being linked. Eric and Keith do a great job of also having iNaturalist records as well. And so those will have a in situ photo. They will have a habitat photo. They might have more exact coordinates. I've run into that a lot too, where the herbarium specimen label has the information. You find it on iNaturalist and it actually has full coordinates. So we have to aggregate it. So that's what myself and the botany data techs do, is we have to go through and we have to find and pull everything together and pull out the unique occurrence information into one record that we bring into our databases. So what are we pulling from SERNEC? Mainly some of our more important things that we grab are the CALIG numbers and the OCC ID, because I'll get into that later. But that kind of tells us what records we've already crawled through and what we haven't. The determination history is really important for us. We also keep track of that. And then we can aggregate that data for tracked species. Collection information, this is really, really important to help us find unique occurrences. So I didn't really go through this before, but unique occurrences are really, really important in terms of Herbaria because there are lots of duplicates. Some specimens have one duplicate or no duplicate, and then there are some of these rare plants are Leonardized, as we like to fondly say, where there are 30 specimens, and so you go onto SERNEC and you search for a specimen in South Carolina, and 60 records come up, and so you're like, oh, well, there's a lot of this plant. There's 60 records. You look closer, and 30 of them are Stephen Leonard's. So we really, that collection information there is really tells us, is it a unique occurrence or not? Avery and I will go in depth in this in the presentation after this, so I'm not going to right now. But location information, we use that for mapping. The occurrence information is obviously very important. We do have, there are botanists who are aware of what they are doing, what they're looking for. And so they will put population counts, which is amazing because that's usually one of the things that's missing off of herbarium specimen labels. And then we do care if it's type, if it's a type specimen, or if there's citation information, which unfortunately there's not a field for that right now on SERNEC. So we do appreciate people who do make notes of that digitally, because as I said before, the paper often has more information. So Darwin Core is our friend because we download the data in Darwin Core format. And then we basically crawl through and we say, okay, have we processed this record before? Yes or no, based on the OCC ID and the catalog number. And if we have, we look to see if the information has changed. If it has, I take a look what has changed and see if our record needs to be updated. But it also lets us find out if there are new occurrences that we need to process or our targeted occurrences. So then we fold the new data into our portal. And so as Herrick said, with this great digitization process, it becomes easier. Some of the fields are standardized to the point where we can pull this in, but we are still working on that because as Herrick said, this portal was made first for fish. So the fields don't always make sense or they might be ambiguous. So from the SERNEC side, what do the botanists do? As Herrick alluded to, in fact, Eric is a prolific annotator digitally. We actually link the SERNEC portal link into our database so that if somebody is looking at the data, they can click on the SERNEC portal and go straight to the specimen. As you all know, phenology students and volunteers really struggle with the phenology side. And so the phenology might need to be updated or they might notice something like that, or they look for data on species we were thinking about tracking and folding into our SC Flora data. We do pull in some data into SC Flora, just all specimen data. We can't parse it because there are over 350,000 records for South Carolina. And unfortunately, a lot of them are missing all the collection information, but we do try to get some idea of distributions of plants based off of SERNEC. So this is one thing I did want to bring up to all of us who have given editing privileges on SERNEC. We really appreciate it. For those of you who haven't, please consider doing so. We spend a lot of time on these records of yours, and so we notice things. And so as Herrick said, it improves your data quality if we are able to transcribe things. So oftentimes if I'm targeting a specific species, the first thing I do just to make my life easier is for all the institutions I have editing privileges for, I quickly go in and fill in the collection information and then go back and then download it. Just to make it easier, but then we can go back and fill in more information. So editing privileges are really, really important to us because we can help you make your data better and it makes our searches easier for us. So this is an example I just wanted to show you guys. So this was Coreopsis integrifolia, which I worked on. Not only did we get the data into our database, but I fed it back to USDH. So these tracked species, we can work on feeding that data back to you guys to improve your data quality and your public display. And I did want to, say Herbaria are more, and especially hearing about Duke, this makes me really sad because Herbaria specimens are great in a lot of ways because they can be physically re-examined. So if there's taxonomic changes, you can go back and you can go into the cabinets and you can look at them over and over. Destructive sampling, as Mihai said, can be used for genetic data and study. It can also be used for conservation. Hannah and I were at the Kansas Natural History Museum, and they were talking about expanding populations of black-footed ferret using museum specimens. It was a bottleneck species that they were able to grab the genetic material from the museum specimens for decades ago to reintroduce genetic diversity. Also, there are seeds that can be used for ex situ conservation. So physical specimens are important. We do not like to see these specimens go far away from us or to disappear completely. And this is just real quick. Herbaria, it's more than just data pulled from online. There are connections with collectors. As Dr. Korn knows, I email him sometimes, like, can you give me more information about the specific specimen? There are connections with botanists. There are maps and other documents, sometimes just forgotten in the box that are really important for us to help you reference. Field notebooks of collectors that have extra notes or more information that didn't make it onto the label. Destructive sampling, institutional knowledge, and also at some point we'll be asking you because USDH has a mysterious flora that just appeared. We don't know who wrote it. If anybody could help identify it, we would really appreciate it. So that is how we pull the data together.
So we talked a little bit about what DNR specifically does with all of this collected data, whether it be herbaria specimens or naturalist records or mystery floras if we find them. Csilla is great at making these smart art graphics. So our South Carolina flora occurrence data gets fed into, it kind of explodes once we incorporate it from SERNEC or our naturalist, explodes into a lot of different products for us. It helps us classify rare ecosystems. We've been going through a big process this summer of developing a new vegetation classification in for South Carolina. A lot of that is actually being derived from herbarium specimens in the habitats that are listed on herbarium specimens. Our rare species list helps us drive conservation priorities. So what were clusters of rare species? What properties contain large populations of those rare species? They help us prioritize what our rarest plants are in the state. So where do we need conservation attention? We can look at something like Camassia psiloides. We have that on no protected lands right now. That should be a big conservation priority. Once we can define what our rarest elements are, whether they be plants or animals, we can actually make conservation happen. We can identify invasive plant species. We can detect new things that are coming into the state. We can detect how frequently those invasive species species are being collected. So we can see if it's becoming more widespread or more aggressive. We can narrow down our data into species, aggregating species distributions and ecosystem distributions. We can rank them. All of these data combine into conservation actions for us, especially with the Heritage Trust Program, what properties we buy. We've been using rare plants to identify what properties to buy, like 40-acre rock or Peachtree rock, for 50 years. There are a lot of other ancillary things that we do with this data for conservation. It's extremely important. Once we buy these properties, we use the data to manage rare plant populations, mapping rare species on each property. We publish taxonomic updates and discover new species in herbaria as well. We're working on a couple of things right now. We'll be talking later about the South Carolina Plant Atlas that we're collaborating on. A brand new invasive plant list, which is in your packets. It was just finished last week. A lot of that is based on herbarium. So statewide plant conservation, we collaborate extensively with other partners to do plant conservation. A huge part of this database that we spend all of our time on is also used for environmental review. So projects come into DNR, fortunately not to me, but go through an automated process. It queries our database. If somebody is applying for a permit to put in a cell phone tower, it comes through environmental review, which flags where rare plant populations are, or wildlife populations too. We're just part of the pie here. And we've also used these data to collaborate on a regional and local level or regional and global level on what our conservation priorities should be, like the regional species of greatest conservation need, collaboration, and G ranks with nature reserve. So a big thing is land acquisition. That's why Heritage Trust was originally founded. So we've got over 70 heritage preserves around the state. That's where Samantha and Eric and I get to spend a lot of our time if they let me go out in the field. But once we buy something, we can't just let it sit there. So we have to manage those properties, and we use our data to drive that. So Samantha and Eric and I are constantly going to the heritage preserves and mapping where all the rare plant species are, so we can make recommendations on how to protect them. Whether that be using prescribed fire, one of our biggest conservation tools, doing other forms of woody plant control if we can't do prescribed fire, like chainsawing down alders in the mountains and mountain fens. Invasive plant control is our other biggest threat to biodiversity. So what populations of rare plants are threatening our ecosystems and rare plant populations. We are also starting to do rare plant introductions and augmentations as well. So putting things back where they used to be based on herbarium specimen data. So here's our network of heritage preserves. We're up to over 72 at this point. Heritage preserves across the state, protecting over 150,000 acres. It's been a tremendously successful program, buying the best natural areas and cultural areas. It's doing it again.
And for you.
So once a property is designated as a state heritage preserve, it's permanently protected from development. And here's an example of kind of the herbarium history and rare plant population leading to action. Does anybody know what this is other than Herrick? Godfrey's privet, Forestiera godfreyi.
No.
It's a species, it's mostly on a 7 or 8 populations in Florida, very few populations in Florida, 1 population in Georgia, and about half a dozen populations in South Carolina. Our Georgia and South Carolina populations are all on Native American shell middens or rings. Entirely, highly calcareous habitats right on the coast, very threatened by sea level rise. This was first collected in Bluffton by Dr. Mellichamp in 1877, who lived in Bluffton. He made prolific plant collections. This specimen is at the New York Town Garden, so he's the first one to collect this in South Carolina. It was not seen again until Dr. Richard Porcher, good friend of ours, found it in 1975 on the South Carolina coast. He didn't know what it was and he mislabeled it. It's a mistake we all make. He thought it was a prunus. So he didn't realize that the Mellichamp specimen was out there. And 21 years later, Patrick McMillan looks at it. It's like, holy crap. You discovered Godfrey's privet in South Carolina and you didn't know about it. But there was this legacy of specimens that Dr. Porcher was able to preserve a specimen that created a record that Patrick could go back and look at. That launched a whole expedition on the coast where Richard Porcher was flying his plane up and down islands to look for shell rings and shell middens to be able to look for this more populations of this plant, and they ended up finding more populations. Once they found it, they realized it's still an extremely rare plant. And we have one property with 3 males. And those males were kind of lonely, so it's a dioecious species. They were kind of lonely. So we worked with Patrick to grow out populations of females, what we think are females, at South Carolina Town Garden. And we were able to plant 7 pistillate plants at the Heritage Reserve to augment the males that were there. So hopefully that'll drive some reproduction in this population. So a long history going back to 1887, leading to a conservation action here. So we want to keep trying to do more of that. One of the big goals of the program now since 2020 is to update our list of what we consider rare. When I started in 2020, we had 326 track species. These are things that the nature, by default, the Nature Conservancy or the Nature Serve classifies as S1, S2, or S3, S1 being the rarest species in the state, or SX, which are extirpated in the state. We don't think they're here anymore, or historical, which we haven't seen for a while, but we think could be rediscovered. We are doing something that not many states have done, and it is entirely dependent. A major part of it is dependent on the herbarium specimens, is doing a whole flora conservation ranking. So we're trying to put these conservation ranks from S1 being the rarest plants in the state to S5 being the most common things in the state, every single one of our native plant species, which is approaching 4,000 species. It's a huge task that Samantha and Eric and I and Csilla, and now Hannah, are working on all the time, especially in the winter when we can't do field work. So we have gone from 326 species to almost about 1,322 species that we consider rare. So our rare plant list has exploded. These are things that are, in general, there are less than 80 populations in the state. This also, by the way, includes some lichens and bryophytes. So we're expanding the number, the kinds of taxa we're tracking as well. Our data, your data, herbarium data, iNaturalist data, also, and our field observations also go into regional conservation work, like a big collaboration that took several years, developing a plant list called the Regional Species of Greatest Conservation Need. So these are the rarest species in the Southeast. It was put together by a collaboration of botanists from Pennsylvania to Oklahoma and Texas. So these are published in this publication. These species of greatest conservation need subsequently formed the basis for the rare plant list that we're putting into the brand new state wildlife action plan, which states are required to do every 10 years. So we are completely rewriting the state wildlife action plan that helps get funding to protect rare ecosystems in each state. So things that were listed on the as species of greatest conservation need in the region were listed by the Endangered Species Act as threatened or endangered or high priorities on our new SWAT. It'll be published next year. Identifying invasive plant species is a huge importance of herbaria. Brazilian pepper was first detected in South Carolina because somebody a homeowner sent in a specimen from the coast, from Hilton Head Island to the University of South Carolina Herbarium. This is one of the top 3 invasive plant species in Florida, and now it's in South Carolina, and we've also detected it on Edisto Island as well. That's the plant I found in Edisto. But we find new populations of invasive plant species, new species of invasive plants in the state every year. So, for example, Ardisia japonica found last summer. And these are all documented and processed in positive herbaria. And we're also seeing we're able to detect trends for invasive plant species. So autumn fern, which a lot of you probably have in your yard or your neighbor's yard. The first detection of this in the wild was actually, I think, by Dixie, who deposited a specimen from Clemson Forest, I believe, a couple of years before John and I were crawling around a property in the Piedmont at the Love Shack on the river. Now it's in a ravine. Now we can't go out in the woods and hardly not find it. It has become so widespread in the state. This is the population in Georgia that Alan Kretzer photographed, but we're able to document how quickly these things are spread by the use of herbarium specimens. Another one of the projects is DNR is working that is the herbaria are critical to is we've got funding from the Fish and Wildlife Service, Ecological Services Office in Charleston to update our whole 50-year legacy of database records for federally listed, endangered, and threatened and at-risk plant species. So that's the collaboration with, where is Avery? Where you are. That Avery is working on going back, and one of the parts of the process is looking at all of the herbarium records that these data are originally based on. So things like Coreopsis integrifolia and Real Trillium and Miccosukee gooseberry is we're working on collaboratively with the AC Moore Herbarium to update all of our data records for this based on newer technologies. So we are involved with Herbaria in so many ways. We feed back data to your portals if you give us editing permissions. You connect Herbaria to resources with mapping resources. Advise on it. We can advise you on imaging equipment as well, imaging standards. We can sometimes help with the imaging itself. We have seated technicians for special projects like Coastal Carolina University, which is reinvigorated an old herbarium, a really important collection, especially for Horry County. So now back to Herrick.
Yeah, I don't want to beat a dead horse so much, but what happened here?
The little bar came back.
Oh, there you go.
Oh, sure. Okay.
Yeah.
All right. Whoop.
I was gonna make it go away, but.
All right. In terms of like mobilizing herbarium data, I think we've alluded to a couple of cases where we've had a collection that was underutilized or sort of in dark storage and recently daylighted. Herbaria have certain advantages over some other collections in that most of our specimens are pressed flat sheets. So we're sort of two-dimensional. We don't have this challenge of getting a good image of the specimen and capturing more than just one dimension. So you can move pretty quickly forward in terms of getting your collection digitized by just taking images forward. That's one of the primary sort of workflows that was pitched as part of the starting effort within the SERNEC project. Was just image capture first, right? Some of the other alternative workflows involve skeletal data capture, where you have sort of a dumbed down form or just a distilled set of fields that are taken out of that Darwin core group of standards. And you'll just enter those sort of rapid fire just to get the specimen record on the map, so it's discoverable. Some of the other efforts involve batch processing. So if you have a spreadsheet that's already got your collection catalog, just a flat Excel file, we can rapidly upload that into the SERNEC portal. It's published online, and then other researchers can use it. And as I said earlier, the data use needs improvement of the data. Csilla, I think, mentioned using duplicate record matching. So if you're doing a skeletal record development methodology, basically you just need a collector last name and the collector number. And those are the primary two fields that it'll do a match off of. And when you put those two little bits of information into something like this aggregated data set that SERNEC represents, it'll go out and look for other records that match those criteria and say, hey, we think this is a potential match. And assuming that that record has been more fully developed at another institution where the duplicate is, you can just migrate all those data over to your data set. The SERNEC environment also has georeferencing tools that'll help you put dots on the map, and this is very important for establishing species distributions and just knowing where the specimen was collected from in a digital mapping environment. And then we had Michael Denzo tell us about Notes from Nature. There's this other offshoot called Bionomia that we haven't touched on that actually looks at more of sort of the biographical information of the collectors involved in these collections. And it is an interesting sort of open source environment that is actually grabbing data from GBIF, which I'll talk about in just a second. But it goes out and looks at all these collectors and it tries to like sort of match up the various iterations of their names. If you've got a certain individual might be entered as Brown, Herrick, or Herrick HK Brown, or H Brown, something like that. It says, I think all these things are the same person. And then it allows citizen scientists to go out and say, oh yeah, that is the same person, and he collected this specimen and that specimen. And so it sort of allows you to attribute all the collecting efforts to any individual collector, and kind of further establishes their contribution to the sort of research. And so that's only possible if once you're adding your data into something like SERNEC, you publish to a data aggregator. That is one of these higher up sort of data environments where iDigBio serves as sort of a national resource. Remember I said SERNEC is kind of a regional southeastern US. iDigBio is interested in the entirety of the US. And then GBIF is a global environment. Once you have your data mobilized into one of these digital environments, particularly like SERNEC or a symbiotic driven system, it's fairly easy to get those data aggregated or published to one of these larger data sets like iDigBio and GBIF. And the point being there is that the more exposure your data have, not only are you advertising your collection and the work that you do, but you're allowing access to other researchers that might not discover it. Say somebody's in China, they're not necessarily going to know to go to the Southeast regional portal to find the information they want. They might discover it on GBIF first. And you can still get feedback from those researchers to help improve your data quality. Okay, this is where that's I thought was going to land on this slide. At any rate, so the same kinds of feedback that you'll get, I mentioned taxonomic annotations, georeferencing. Again, I think this is something that DNR is focusing on a lot. A lot of our collections pre-1974-ish or so don't have any coordinate data. It wasn't until like mid to late 70s that you really start seeing latitudes and longitudes showing up on the records. And so in the more recent cases, it's fairly easier to georeference stuff because you can just take the coordinate values that your original collector gave you. But in the older stuff, you kind of have to triangulate and use the verbal description of where things were collected in order to kind of approximate where these things were on the map. And a lot of times having the data out there can help you improve the quality just because you might have an image of a specimen that's matched with the wrong record, right? So, technological failures happen all the time. And when somebody's looking at it and say, well, this thing says it's Helianthus egertii, but it's actually Coreopsis integrifolia or something. And somebody finds this mistake, they can easily send you a note either through the portal or they can just email you with your contact information on the collection profile and let you know that you need to fix something there. And then there's other horribly egregious errors that need correction. Sometimes you have just complete utter nonsense, or it may have been a case where somebody was using a feature like carry the data forward. And that's where you enter one record and you say, go to the next one, and it just carries everything forward. And if you have a lazy data technician, then everything is from Horry County, South Carolina for some reason, even though it's from Florida. It'll help you discover these sorts of things once you get people looking at your information. Okay, and so it's both sides of the street. Okay, so, as agencies like DNR use our data, they're giving us, positive feedback and helping us improve the data. And then sometimes we have things that turn up in Herbaria that, just weren't really a big deal at the time when they were first collected. I'm thinking like, Herbaria cylindrica, it was like the, Kogar grass. It's a huge priority now because it's such a pest in terms of like its ill effects on native ecosystems. And I think the first time it was kind of on the radar for Department of Plant Industry was, I think the first collection they thought was, John, you remember? John had collected this thing like 10 years before the folks at the Department of Plant Industry thought it was first found in the state.
1987.
Yeah, yeah. So it's like, they had it on their benchmark as being in the 1990s sometimes at any rate, these vouchers might be in there, and once we have them published and discoverable, they can help inform, management decisions that are coming out of this. So I'm gonna go off script a little bit right now, because I had a few more slides that I just discovered. And I want to talk about some of the, whoop, how did we get that far? Wait. Let's see. So some of the use case scenarios where Herbaria really helped with DNR efforts were USC Upstate. They did an image forward workflow. They captured all the images and then they developed skeletal data. And we were alerted by Upstate Forever, the property near Travelers Rest that was slated for development, and it was right next door to one of our heritage preserve properties. That was known to harbor two federally listed species, dwarf flowered heartleaf and bunched arrowhead. And I knew, Jill Newberry was up there at USC Upstate, and I knew she had done a bunch of work with Hexastylus, so I just kind of like, started searching through USCS's Hexastylus collections, and she would have a map with a dot right where she collected it on there. And sure enough, there was a sheet. It didn't have any of the location information digitized except for what was visible in the image. And sure enough, there's Hexastylus naniflora collected right on this property in question. So it was a case where DNR was able to use this voucher material to help prioritize the property for acquisition and preserve that population, which also helped maintain the integrity of the adjacent property that was already there. Furman University had, as the only record, I think, of Platanthera integralabia. I don't think it's a common name for that. So, it's an orchid that's up there near Jones Gap, and Sarracenia purpurea variety Montana, which is one of the few extant populations of purple pitcher plant that's up in the mountains. So those are the few vouchers that we have from that location there. They're basically unicates. They're unique specimens. There aren't any others like that in existence. Appalachian State University with the Ives Herbarium had some collections of short Glacifolia, we call it Oconee bells, right? That are from places that are now inundated, right? You guys see gorgeous area. And then there was another interesting case where the small community college, Catawba College in North Carolina, was brought to my attention through a social media post by Wesley Knapp, who's now kind of a bigwig with NatureServe. And he said, yeah, I went to visit my old alma mater, and the curator there showed me the collections, and we were going through there because we knew he had some unique stuff. And I was like, we can digitize that. We can get that stuff out there so people can use it. You don't have to do the herbarium crawl. And like in a week's time, it went from no existence virtually whatsoever to having several thousand records online. I mean, it happens that fast. It really puts the collection on the map. And when you're talking about trying to argue the merits of a collection like this in terms of an administration, being able to document how those collections are used helps a lot. Okay, so get back on track here. This is looks like a bunch of minnows swimming in eddies or something, but the sort of circular pattern you want to focus on is really sort of the reddish colored areas in the center that represents some of the workflows within the herbarium pipeline, where specimen data are published online once you've taken an image and started developing that metadata that goes with it. And then you have agencies like DNR that might pull some of the information for their studies. You might also have some parallel work being done by researchers that are looking at, floristic types of information, trying to define plant communities or do taxonomic research and that kind of thing. And each one of these sort of spin-off environments has its own sort of workflow where they're developing data and feeding it back to the home institution. So as annotations are applied, we get that information, we can apply it to the physical specimens. The DNR partners with a bunch of other agencies. So in fact, with this little eddy right here, you've got several other spinoffs where they're actually sharing data with U.S. Fish and Wildlife, with NatureServe, and those sorts of partner agencies. And then the data are coming back into their pipeline and then back to the herbarium. And ultimately, that workflow involves some additional change to the physical collection where we're trying to document all how that information has come to us and what changes need to be made. So then we need to re-image the specimen and you go into this little wheel over here. And then finally, I would like to touch on something that's been kind of I don't know, a little bit of a buzz in the natural history collections community. Extending the digital specimen or the digital extended specimen. It's sort of this phrase that was agreed upon between the United States Natural History Collections managers and colleagues in Europe. They couldn't really agree on like these concepts, and I'm sort of on the fence of what sort of terms we really should be using. But basically, the take-home message here is that natural history collections are really terrible at documenting the environment. If you think about it, the specimens that we curate really are kind of like a little part and parcel out of an entire community. And we're trying to sort of make order out of the chaos that, the natural world that surrounds us by identifying a specimen as this species, and then we catalog it into this little cubby hole with things that are similar in appearance and that sort of thing. And so we kind of lose some of the little, the stickiness of the web of everything that's like, brought together in nature. And what we're trying to do now is like reestablish that. A long history of trying to treat these collections as just a taxonomic collection. And now what we're trying to say is, oh, well, this collector was at, point X on the ground at year Y or whatever. And he not only collected that specimen, but 20 others. And from that information, we can kind of get a snapshot of what the forested community was at that point in time. If we look at them just as individual specimens, you don't necessarily see all that information. Now, nowadays, people are a little more detailed with the kinds of information that they record on their labels, so you don't see that as much. But remember, we have 300-year legacy of not recording all that stuff. So now what we're trying to do is this extensions of the digital specimens and reestablish those connections. And that can include things like publishing information to GenBank, bibliographic citations, or Exicati, where there's sort of bound volume of specimens. We can add in situ photographs. So in addition to the distorted, flat pressed specimen, we can have like shots of what this thing looked like in life. Micrographs, so we can capture really close-up detail of some of the like 2 millimeter plant hairs that are very diagnostic and differentiating certain species. And then adding the biographical information that I mentioned earlier with biography. And so along those lines too, you can also kind of map where collectors were in the world and what their sort of collecting habits were. Sometimes I know I've seen like some collections by Radford where he basically was just on a road trip trying to collect every dicanthelium on the side of the road. It was like not like he's going to a really cool place like Stevens Creek. He's just, trying to figure out how this group of grasses is different from one another. And then other things that we are trying to do now is sort of phenology. This plays into sort of a global climate change sort of thing where we're looking at trends and when things are flowering and fruiting and trying to explore those traits and those things.
Ready for questions?
Yeah.
Go for it.
How difficult is it to put in good data for these older specimens and you want that latitude, longitude in there? Because I've seen some records where they've got the 6 decimals and I'm like, there's no way they can have that from a specimen collected 100 years ago.
I am going to talk about that in the data standard.
Oh, okay, okay. I'm just introducing you.
Yes, actually, that comes up a lot. I hope I have the comic in here. I had a. Do I? If not, I will pull it up next week.
I don't think, unfortunately, that we did not put the collection, I don't remember who did this, but it was something like. All they use as their descriptor was soil. And no locality information.
There's a specimen that I'm working with right now, the type specimen for the species.
Mhm.
Just says, it was a Michaud specimen, it just says something like west of the Appalachian Mountains and Kentucky. That's like the whole, that's
Yeah.
the locality description for the type.
Yeah, I would like to point out, I did not have that in my presentation, but yes, there is a certain amount of precision, and then, you know, if you put all those things, you copy it from Google, you're either handing out raw floating point variables, or you built a database to track individual atoms. Either way, stop. So, typically it's like, I think it's, what is it? It's, 4 is what we usually go for.
Yeah,
Because anything beyond that doesn't really make any kind of sense.
I've gotten coordinates and then gone back to the site and like on the Appalachian Trail and you're off the trail somewhere and you want to find the plant you saw 2 years before. And 5 digits puts you within about 10 feet of it.
Yeah, that's actually a really good point. It's amazing, and also putting which way you're facing at the coordinates helps a lot. So when you get to the coordinates, you know which way to look on the trail. The trail, so. You need to. Yeah, just use it. I was just trying to make sure I didn't take the full projector down with me. Okay. Oh, okay. All right, so those of you who have attended the SC Consortium meetings last year, thank you. Those of you who haven't, you are always welcome. I've received feedback saying, oh, well, I thought that, you know, since I didn't attend the first one, that I couldn't attend the rest. No, these are all